So thank you so much, Michael, for having me and for everyone for tuning in and just listening to our talk this morning. I'm going to be talking a little bit about how I think about cognitive resilience and how our lab tries to use lifestyle behaviors to try and get at some of the biological pathways that might underlie cognitive resilience. So just to jump in, I think we've all seen slides like this before, but this is really the problem that we're trying to tackle. So this is the development of cognition with age. And so these are real data from the NIH toolbox. We can see with age, in early childhood, we see rapid gains in cognition that kind of peak around age 30 or so. And then we see pretty precipitous increasing decline with increasing age. And so you can find yourself on this line, but this is really the area where we're the most interested. This is sort of the natural development of cognition. How do we help support this line to become more flat and less curved? But I would say that this line is really not the whole story. And in fact, there's a lot of heterogeneity around this line. So if we actually map out the individual data points here, you can see there's a ton of people above and below this line. And this is what gets me really excited, is to try and leverage these individuals. So if we zoom in a little bit further, we can see that there's several individuals who are in their 70s and 80s over here, who are performing exactly the same as their peers who are 40 or even 20 years old. And so the question to me is, who are these people? What's their kind of special sauce and what keeps their brain going? And how do we try to bottle that and use that to treat neurodegenerative disease? And just to highlight, again, how we're thinking about this even more clearly, we have these are real participants in our research study. We study typical cognitive aging in our lab. And so this is, on the left here, we have an 81-year-old woman with 20 years of education. And on the right, we have a 74-year-old man. And you can see that on imaging, the woman here, she has, a fair amount of atrophy. It's asymmetrical. She has more left than right atrophy. Her hippocampi have some, pretty significant gaping holes there. And if you look at her flare, she also has some fairly significant white matter hyperintensities, both punctate, but also becoming confluent. So pretty significant white matter disease. On the other hand, this gentleman has pretty full brain. His hippocampi are nice and plump. His sulci are nice and tight. On flare imaging, you don't see too many white matter hyperintensities, just a little bit of this periventricular capping. So what do these people look like? So this woman on the left is actually diagnosed as clinically normal. On the MMSE, Global Cognitive Screener, she gets 29 out of 30. On a list learning task, she can recall 15 out of 16 words after a 20-minute delay. And she's really speedy on a trail making test. Whereas this gentleman on the right-hand side, he's actually diagnosed with mild cognitive impairment. He's a 24 in MMSE, so impaired. He's not able to recall any words on the list learning task after a 20-minute delay, and he's quite slowed. And so, what is it that's supporting this brain on the left here, despite even having some pathological hallmarks that's not present on this brain on the right? So to, again, wrap our heads around this a little bit more, I'm going to walk through a model that I've been thinking about on how to think about cognitive resilience and reserve. I think these are terms that we're all trying to grapple with and work out right now. And so this is certainly a working model, and it's been adapted from the previous works that have been done by Aranaza, Urquillo, and Vermuri, as well as Tom Montine's group. I've also chatted with Tim Homan a bunch about these types of models, but I'll say that this one only represents my beliefs, and we can chat more about it later. But just briefly, this is the resistance and resilience framework where we have these exposures, and these are sort of considered risk factors that would put someone at risk for a pathology. And so if someone has one of these exposures but does not have the pathology, they might be considered resistant. On the other hand, someone who has a pathological lesion but is functioning quite well might be called resilient. And this sort of dotted line on the top here is to acknowledge that this is likely a highly dynamic feedback loop. So once someone is being resilient to a certain pathology, they might actually be resistant to downstream pathological processes. So to make this a little bit more clear, I'll walk through an example. So thinking about age and amyloid, for example. So age would be our risk exposure. As we get older, we expect brains to have amyloid in them. So an 80-year-old that has very low levels of amyloid would be considered resistant to amyloid. On the other hand, an 80-year-old with high levels of amyloid, but who is functioning quite well, would be called resilient to amyloid. And perhaps in this individual who's being resilient to amyloid, they're actually being resistant to tau. And so you can think about how this cycle might continue with lots of other biological processes. So I think the biggest question for us is, what's in this black box here of resistance and resilience, or green box as the case may be here? What are the mediating factors? So I'm just going to overlay some of the other terminology that I think we commonly hear. So I might argue that these sort of neurobiological capital, or one's capacity, sometimes referred to as reserve, might mediate this resistance ability. So the maybe genetic abilities or abilities that have been developed over a lifetime may be particularly important for mediating the presence of pathology. And on the other hand, in the context of resilience, once a pathological lesion is present, there may be important accommodation and plasticity phenomenon that needs to work around this pathological lesion in order to maintain skills and maintain resilience. And of course, these two phenomenon are likely highly dynamic. Someone with high neurobiological capital likely has a greater capacity to accommodate for lesions and back and forth. And so again, in my work, I'm really interested in trying to understand the biological pathways that might be supporting resistance and resilience. So to simplify this even further, this is really how I think about it. If we could find the resistance molecules that prevent disease and the resilience molecules that help us live with disease, these are our golden tickets. This is exactly what we need to find to cure dementia. But easier said than done. I think we'd all love to find that. It's quite hard to do. And so the way that I've thought about it is taking a step back and thinking about, well, what factors are associated with resistance and resilience? And so we know that lifestyle behaviors, things like physical activity, cognitive enrichment, social engagement, are very consistently associated with both resistance to pathology and resilience to pathology. And so if we can figure out, in my head, the biology, the neurobiology that's underlying these lifestyle behaviors, these might be the same molecules that are going to support resistance and resilience moving forward, and could be therapeutic targets. And so this is really the framework that guides many of my studies. Happy to chat more about the model moving forward. So I'm going to jump into some data, and let's see, I think, it's hard to open an aging journal at this point and not see something about exercise or lifestyle. So I'm not going to belabor the point. I'm just going to highlight one of our recent works just to hopefully further drive home the point that we think that lifestyle really does shape our brains. So in this study, we followed a cohort of individuals who carried the autosomal dominant, one of the autosomal dominant genes for frontotemporal lobar degeneration. This is the all-FTD study. And so in this study, this means that the mutation carriers, if they live long enough, they will get FTD. It's sort of the perfect nature versus nurture design, where we can test questions around lifestyle and how much does environment matter. And so we asked these mutation carriers at baseline, how often do they engage in physical activity and cognitively enriching activities? And we found that kind of excitingly, that the gene carriers who at baseline reported greater physical or cognitive activities demonstrated about 50% less decline on the CDR sum of boxes year over year. So more active lifestyle, slower functional decline, which was exciting to see. And really, furthermore, we found that if we measured their frontotemporal atrophy over time, that the gene carriers who were more physically and cognitively active demonstrated about twofold better performances on our cognitive tests for their frontotemporal atrophy compared to their less active peers. So it seemed like they were kind of outperforming the sort of hallmark pathological marker of frontotemporal dementia when they were had more active lifestyles. So I think, again, this sort of supports, there's issues of reverse causality. Obviously, this is an observational study, but I think this helps to support this proof of principle that there's something around maintaining these active lifestyles and using our environments to help shape our brains, even in those with very high risk. And so next, how can we get at some of these factors? What's the biology? What's driving better brain outcomes, cognitive outcomes when we engage in these behaviors? And so I'm going to walk through a really fun collaboration that was recently published with Saul Vieda's group here at UCSF. So just bear with me as I go through some of the experiments. So in this study here, Saul and his team, we were interested in physical activity and the biological factors. And so what we did is he took aged mice, he exercised them, and he sort of showed the common effect that when mice exercise, even if they're older, we see neurogenesis in the hippocampus, and it's correlated with better behavioral and cognitive performances in these mice. So this is great. We've seen this previously. But the question is, what's going on here? How does this neurogenesis happen from exercise? And Saul has, extent experience on systemic factors and using heterochronic parabiosis and blood factors to try and rejuvenate the brain. So the question then became is there something in the blood of these exercise mice that might be neurogenic? And so what they did was they exercised some old mice, and then they injected the exercise blood into sedentary old mice. And they found that, yeah, actually just from the exercise blood, we can recapitulate this neurogenic effect, as well as the behavioral benefits. So exercise blood seems to have something special to it for the brain. So then the question was, well, what the heck is in the blood? And so he exercised some old mice, he exercised some younger mice, and ran some mass spec to try and identify the most potent overlapping exercise factors that we could find in the blood. And there were 12 individual factors that came out as sort of overlappingly increased when the mice exercised. And he decided to target this one molecule called GPLD1. We can sort of get into it in if there's questions around it, but the main framework around it was, well, first of all, we actually don't know very much about this molecule, so it might be sort of a novel, important factor to go after. The other interesting thing about it is it doesn't necessarily increase with age. It seems to be an exercise-specific factor, and so that made it kind of an exciting target. And so what if we took this one factor, GPLD1, inject it into our old mice? We actually found that if you just inject this one single molecule, we could, again, recapitulate the neurogenic effects of exercise. So we sort of boiled exercise down in some ways to one molecule in mice. But then, of course, I'm a clinical researcher, and I think the mice studies are incredible. But what does this mean for humans? And so we have a Fitbit study at the Memory and Aging Center. And so we took some of our healthy older adults and took the ones that had the most physical activity and had the least physical activity from the Fitbit. And we measured this one molecule, GPLD1, and we did find that in the healthy elders who had more physical activity had higher levels of this molecule, suggesting that there are some translational benefits potentially across mouse to human. And I'm not going to go into all of the experiments that they did. The paper is really incredible. I encourage folks to look at it. But what is this molecule? So digging into a little bit further, it seems that GPLD1 is primarily expressed in the liver, and it's increased with exercise. It's really not as highly expressed in other organs. And they did some pretty cool sort of high bit tagging and found that it does not seem to readily cross the blood-brain barrier. So this is, again, the expression in the liver compared to other regions in the brain, and it's just at very low levels. So we don't think it's necessarily sort of directly targeting the brain. So then what might be going on? Again, doing some mass spec experiments, we found that if you inject mice with GPLD1 and take a peek at their mass spec proteins that are increased after the GPLD1 increase, and compare it with some of the processes that were increased in the exercise mice, there was a real convergence of pathways. It seemed that coagulation and particularly complement cascades might be triggered by both GPLD1 and exercise. So the sort of working framework that we're thinking about this is that exercise sort of releases GPLD1, which decreases complement in the periphery, and it allows for lower levels of complement in the brain, which we know it could be a helpful thing for our astrocytes and our microglia to help maintain synapses and brain health. And so just to, again, take a step back, I think using these types of tools, again, leveraging exercise to get at biology, we've found that, there might be blood components from exercise that can rejuvenate the brain, and perhaps even a novel liver to brain access that we haven't been thinking about previously, that was discovered through this work. And maybe GPLD1 in and of itself could be a target more broadly for age-related cognitive resilience. And so I think we're going to try and follow up with this molecule and others, and other clever ways to approach lifestyle to get at the biology of resilience. And so shifting back to humans, which is most of what my work is, the other question that kind of irks me and in our work is thinking about personalized medicine and precision health, and well, which biological pathways of resilience are important for which people. And we know that sex is really important for cognitive aging. There's different phenotypes as individuals age across sexes. We also know that many of the cardiovascular risk factors differ by sex. And there's also sexual dimorphism in the immune system. And so we had the question of, how does physical activity benefit neurobehavioral outcomes differently across sexes? So in this study, we took a cohort of, these are typically aging older adults, so healthy folks. We had 45 males and 45 females, and then we matched them on age and education. Often these demographic factors differ between the sexes, which I think can complicate things when looking at sex differences. So this is our discovery cohort, and then we also had a replication cohort of unmatched male and female, typically aging older adults. And everyone completed a self-reported questionnaire of physical activity called the PACE, Physical Activity Scale for the Elderly, as well as blood plasma that was measured on the Mesoscale Discovery platform for 11 cytokine markers. They completed neuropsych eval as well as a brain MRI. And so looking at the inflammatory outcomes, we ran a multivariable model entering each of the 11 cytokines in as the outcome. And we found that there was a significant physical activity by sex interaction for the inflammatory proteins. And so digging into them more individually, I highlight this data not necessarily to go through every figure here, but just to show the consistency of the effect. So in each of these figures, on the x-axis is physical activity, and on the y-axis is one of the cytokine markers. The blue line is male, and the red line is female. And you can see that across each of these figures, the blue line here that the more men exercise, the higher levels of physical activity they have, the lower their inflammatory cytokine. And it's pretty consistent across each of these cytokines. Whereas for women, the line is much more flat. So exercise did not relate very strongly to physical activity in women. And we thought this was, just remarkably consistent, especially given human data. These cytokine markers relate to one another, but they're not collinear. They only relate about a correlation of 0.2 or 0.3. And we replicated this in the replication cohort, and we found that these specific proteins demonstrated the most consistent sex-specific effect of exercise. And we also looked at cognitive and brain outcomes and found there was also a hint that males showed a closer relationship between physical activity and brain volume, as well as cognition. But those models didn't replicate as strongly in the replication cohort. So then the question was, well, what is it about these peripheral immune factors that might be relating to the brain in men compared to women? And so what we did was we took these three proteins that were showing the strongest sex-specific exercise effect, MCP4, MIP1 beta, and eotaxin, and created an inflammatory composite from them. And then we related them to the brain. And so what we're seeing here is on the x-axis, we have this inflammatory composite using those three activity-related proteins that showed sex-specific effects. And then on the top here, we have total gray matter volume, hippocampal volume, visual memory, and processing speed. And you can see again, the males in blue, that the more inflammation the males had, the poorer their brain volume, as well as the poorer their cognition. Where in women, these inflammatory markers did not track so closely with their brain structure or cognition. So if we were to sort of put all of those models together, we're suggesting that there might be a male-specific pathway between physical activity and brain health, such that we're seeing that men who have more physical activity have lower inflammation, and men who have lower inflammation have better looking brain structure and cognition. So I think this begs the question, these are observational data, and we need to figure this out through experimental manipulation of exercise and see if we also see this effect. But the question begs, are females benefiting less from physical activity, or are males benefiting more? I can go into more detail, but just briefly, we know that there's, again, sexual dimorphism of the immune system, where males seem to be more at risk immunologically. So women in general mount a larger, more robust immune response. They have faster wound healing. They create more antibodies to vaccines. And so maybe what we're seeing here is that men are really sort of needing physical activity to help maintain that homeostasis of their immune system that's maybe not needed for women. On the other hand, there are some animal models showing that women or female mice that have estrogen loss may not sort of capture all of the benefits of physical activity in the brain. And so perhaps there's some hormonal factors at play as well in older adults. I think this is a really exciting area of work to try and think about the sort of precision of both lifestyle behaviors for brain health and the biologies that might support resilience differently across sexes. It's likely going to be, pretty complicated interplay among chromosomal, hormonal, psychosocial, immunological factors that we'll have to carefully tease apart. But just a hint that we're seeing some differences across person-specific factors. And I'm just going to shift gears now to talk about a biological pathway that I'm particularly interested in. And it's the synapse. And this is something we're trying to tackle in order to understand resilience a little bit more. So why the synapse? So we know that synaptic functioning tracks better with cognition than amyloid or tau or even neuronal cell loss. There's some really beautiful, elegant work from the early 90s from Maslia and Terry and Dukoski showing this. So the synapse is really the biological seat of cognition. And more recently, there have been some really nice case control studies of clinical resilience to Alzheimer's pathology. So in this study from UAB, they took adults at autopsy. They had those who were normal at autopsy and did not have AD pathology. They took those who were normal, clinically normal at autopsy, but did have significant AD pathology. And then those who had clinical dementia due to AD, as well as AD pathology. And they found that one of the major differentiating factors in this resilient group was their synaptic shape and number of dendritic spines. So they found that the dendritic spines where the synapse lives were more elongated. They had more of these sort of plasticity related spines. They're thin spines over stubby spines. And this was a critical factor that differentiated these groups. And the synapse has also shown up to be a unique factor in resilience, and two other really beautiful case control studies of AD resilience, where both of these studies have identified synaptic proteins that uniquely differentiate, that are increased in the resilient group compared to the dementia group. And so on the other hand, we also know that the synapses track really closely with behaviors. So for example, Marion Diamond's work, which dates decades ago, has taught us that an enriched environment, more cognitive stimulation, more physical activity, and social engagement results in better looking synapses, greater density of the synapse, more perforated synaptic heads, more connections. And so thinking about the synapse as sort of this juncture, this converging pathway that might help support resilience on the one hand and be very malleable by lifestyle or particularly or even druggable by pharmacological approaches. But the synapse is hard to get to in humans, which again is what I study. So we've kind of lagged behind. How do we study this in living humans? So increasing technologies, both molecular PET, but also cerebrospinal fluid, have allowed us to start to peek into the synapse in real time. And so I'm going to show a little bit of data looking at a couple of synaptic markers from the cerebrospinal fluid. So in this study, we looked at a postsynaptic protein called neurogranin, and we measured it in clinically healthy older adults. And we found that neurogranin, again, this postsynaptic protein, related more strongly with memory, a functional outcome, significantly and more strongly than all of the traditional AD biomarkers, none of which correlated with functional status, suggesting that neurogranin synaptic protein might be particularly sensitive to the functional status of someone. And in this next study, we were able to measure a larger panel of synaptic proteins in a different cohort of folks. They are also clinically normal. This is in the Hillbloom Aging Network here at the Memory and Aging Center. You can see their demographics here. And this is the factors that we were able to measure in the cerebrospinal fluid in collaboration with the Swedish group. So Henrik Zetterberg and Kaj Blano's team measured these proteins for us. We measured synaptotagmin 1, SNAP25, GAP43, also called neuromodulin, as well as neurogranin. You can see what their functions are at the synapse and sort of where they sit in the synaptic compartments. And so if we look at just the classic amyloid and tau in these healthy folks, you can see more abnormal amyloid, the more tau they have. But once we start to model in the synaptic markers, we found that there was actually a significant interaction between the synaptic markers and amyloid on tau. So in these figures here, we see amyloid on the x-axis and p-tau 181 on the y-axis. And for the synaptic proteins, higher levels are actually associated with poorer cognition, poorer brain outcomes. And so we see that in those with high synaptic protein levels, this is for SNAP25, synaptotagmin, neurogranin, and GAP43. So those with high synaptic protein in their CSF, we see the kind of expected relationship between amyloid and tau, a nice tight relationship. However, in those with low synaptic protein in their cerebrospinal fluid, we see a significant attenuation of the relationship between amyloid and tau, such that there was really no relationship between amyloid and tau in those with more normal looking synaptic protein CSF levels. This sort of suggests that, well, first of all, you might need both amyloid and synaptic dysfunction in order to have the tau aggregation. And or we could think about it as if we're able to maintain synaptic levels to these more normal levels, that having amyloid, does not necessarily have to result in sort of pathogenic tau accumulation. These are observational data, of course. And the other juncture that we looked at is between tau and gray matter volumes. So we were interested, how does the synapse play a role between tau and a neurodegenerative outcome? You can see there's sort of a curvilinear relationship between tau and medial temporal lobe volumes here, though, with those with higher tau having lower medial temporal lobe volumes. But once we start to model in the synaptic proteins, we can see that, again, there's a significant interaction such that those with high synaptic protein in their CSF show the expected relationship between tau and medial temporal lobe volume. So more tau, smaller medial temporal lobe volume. But those with lower synaptic protein in their CSF, they have a significant attenuation of this relationship between tau and medial temporal lobe. Again, sort of suggesting that in those with lower synaptic protein, we don't see neurodegenerative markers, even with increasing tau. So these data might suggest that, synaptic state, synaptic functioning might be central to the development of AD proteinopathy. We've studied this in people who are, quite high functioning, clinically normal. And so we see these relationships, even in these states where folks were not showing any cognitive symptoms. And so we think that this might be a pathway that we want to pursue to target preserved resilience to AD proteinopathy in particular. That's starting to show some exciting signal. How am I doing on time? Okay, great. So I'm just gonna walk through some of the major take-home messages, and then I'm happy to answer any questions. So again, as a neuropsychologist, I was trained that, our brain is what creates our behavior. Our behaviors can be localized to the brain, and this is what we always are trying to figure out. But more and more, I've appreciated the cyclical nature of this, where our experiences are really shaping our brain moment to moment. After this talk, your brain has fundamentally changed, maybe in some ways, and there's a sort of biological pathways that might be that we might be able to tap into to help support better brain shaping, better brain health as we age. And so by using these lifestyle behaviors, we might be able to, hopefully I convinced you that we might be able to get at some clues to resilience, and especially the biology of resilience. So perhaps the molecules of lifestyle might be these molecules of resilience and resistance. And by taking this approach, we can identify novel pathways like a liver to brain axis that we had not been thinking about previously, as well as sort of sex-specific pathways or person-specific ways that resilience may be manifesting. And lastly, that we're really honing in on the synapse, and this could be a really important converging point of resilience. If we can support synaptic health, might this be a way to support cognition, even with all of the pathologies that come with age? Maybe all we need is a synapse, and how can we support that as best as we can using lifestyle as well as multimodal pharmacological approaches moving forward. So I'd love to chat with anyone else who's also thinking about the synapse in living humans. And so just lastly, I just want to thank, huge thanks to the Memory and Aging Center who have supported this work during this time, particularly my mentors, Joel Kramer, who's one of the most collaborative folks that I know. So if you're interested in any of these data, please reach out. We have a large cohort of healthy agers that we are happy to collaborate on. My other mentors, Christine Yaffe and Bruce Miller, are really fundamental in this work. Our whole aging network lab, the B.R.A.I.N lab, as well as our collaborators over at Gothenburg, and of course the participants and family. And I'm happy to take any questions.
Thank you, Kaitlin. That was great. It was really a very exciting overview. So please come up with your questions. So maybe I just start with a question. Have you thought about introducing physical activity training and then look at those changes? And would you expect that short-term interventions like over a few weeks or a couple of months will really have an impact on synapses or even on the immune profile that may significantly alter the risk of developing Alzheimer's disease pathology?
Yeah, this is a great question. And so I actually have a K23, an NIH award to try and tackle this. So we have a behavioral intervention right now where we randomize older adults, healthy older adults, to either a physical activity arm, a cognitive activity arm using a cognitive training platform, or a control arm. And our primary outcomes are these biofluid markers, these molecular markers. We're going to try and get at synaptic markers in this cohort, as well as immune and vascular molecules. And I think if we lean on the animal literature, these processes occur very quickly. So, engaging in these types of behaviors, exercising, et cetera, can, upregulate in a matter of hours, but certainly weeks. So I guess the answer is we don't know in humans. My intervention is a 6-week intervention. So it's fairly brief in terms of how interventions go. So I guess we'll be able to try and tackle that question hopefully soon, and see if we see these biological pathways start to upregulate or downregulate with these behaviors. Though, of course, I think it begs the question of durability. So if you have a brief intervention and you see these pathways upregulate, we don't necessarily know how durable that is if it's a brief intervention in particular. And I'll also say that we collaborate with other folks who have done behavioral interventions, and have captured blood or cerebrospinal fluid. So if folks out there have behavioral interventions that they've completed and they'd like help thinking about their blood, this is a way that we've also been able to collaborate, and we're hoping to come out with, again, some more results to show that once you manipulate these behaviors, you see the biological changes.
Mm-hmm. Cool. Great. Any questions? Please go ahead. So would you expect specifically these changes for physical activity, or do you think that cognitive stimulation, may have also effects on these kind of synaptic or even immune markers?
Yeah, I think the synaptic markers, ideally, if we sort of replicate the animal models, we should see synaptic changes with physical activity or with cognitive activity. And this is work that's been shown, in for decades in animals. But I don't know about the other pathways, and I can see a place where either physical or cognitive activity, might shape our immune systems. I think it's more likely that physical activity will be related to the immune system. There's more data supporting that just so far. But I think there's certainly a case for maybe not the peripheral immune system more broadly, but certainly we know that microglia change in response to enriched environments. So if we're able to detect that, then I think we should see that. That's definitely one of the pathways that we're thinking about.
Mm-hmm. Good.
I have a question, perhaps. That was a really nice talk. Thank you for sharing all those data. I was just thinking about your original slides on the structure of resistance and resilience that you posed as a framework, and then taking some of your data that you shared, and in terms of maybe personalized medicine or targeted interventions. Do you think from your data or other literature that some individuals may benefit in more of a resistance model from exercise, for example, compared to resilience, where some individuals may benefit in both of these mechanisms? And if so, do you have an idea of who those individuals may be? Is that something you're trying to, going back to this framework, explore in terms of why they may be beneficial and in which ways? Because I think the literature is very heterogeneous in terms of, is it resistance, resilience? Is it both? Is it different between individuals?
Yeah, I think that's such a great question, because we know, and I don't know the answer, but I can speak a little bit to it. But we know that these, physical activity doesn't work for everyone. There have been negative and even adverse trials in people with dementia, for example. So what is going on there? And this is where I think we have to figure out this personalized medicine approach. We have a little bit of data from our team led by Molly Memel, who looked at different AD risk factors and how it might moderate the effects of physical activity on longitudinal brain and cognitive outcomes. And she found, kind of interestingly, so if you look at ApoE and amyloid PET at baseline and their physical activity at baseline, that it's a little bit counterintuitive, but so the people who are ApoE4 and have amyloid, they showed the greatest benefits from physical activity on their brain and cognitive outcomes. They had, better looking trajectories. But on the other hand, the people who are ApoE4 negative and amyloid negative also showed the best benefits. So it was sort of like the highest and lowest risk groups. And people who had some amyloid in their brain, we actually kind of saw a hint that if you have amyloid in your brain and you're ApoE4 negative, that exercise was not helping them. So, and maybe, the trajectory was a little bit negative even. So this, I think, begs this question that depending on what your composition is, what your genetic risk is, what your state of your brain is, do you have a significant amount of pathology or not? These behaviors might function very differently. So in the case of someone who is ApoE4 negative, exercise might function as a resistance tool. So in preventing amyloid and being beneficial there, whereas in those who were E4 positive and had amyloid, that may have been more of a resilience mechanism. So maybe completely different biological pathways downstream. So these are data we're trying to replicate, and they're kind of complicated once you get into these sort of personalized medicine approaches. But you raise a really great point. So.
Thank you.
Yeah, thank you. That's something definitely I've been thinking a lot about. And I think Jill Morse recently put out a paper also on AD type risk factors that are affecting the relationship between physical activity and make cognitive outcome and stuff like that. So, and I think it was similar factors like ApoE4 and age and stuff like that that you had mentioned.
Okay, perfect. We'll have to make sure, see if our results are comparable to that. Thank you.
So do you think vascular factors may play a role?
Yes, I think the vasculature is obviously so critically important. I think, but I don't think that's the whole story. Also, I think a lot about the glymphatics, especially in the E4 carriers, and how there's breakdown of the blood-brain barrier in E4, and wondering how, you know, these clearance mechanisms, which we know are related to physical activity, might be bolstered in people who maybe are at risk for poor clearance. And the vasculature is a critical piece, obviously, to that clearance from the brain. But whether it's just sort of a truly vasculature, leaky blood-brain barrier phenomenon where exercise is bolstering the structural integrity of the vasculature, that might be a whole other pathway for certain individuals as well.
Yeah. Okay, anybody else? There's one in the chat function. Ah, okay. So Stephanie just gave a reference, I think, to the
Oh, great. Thanks,
yeah,
Stephanie.
to the article she mentioned. Okay, so if there are no further questions,